A very good morning and a warm welcome to all participants. The theme for today is Biomedical Museum IMR Malaysia, Introduction and Research Collaboration. Our presenters today are from the Biomedical Museum Unit of Institute for Medical Research. I am Shamini N. Sampson, a medical laboratory technologist, also from the Biomedical Museum Unit of Institute for Medical Research, and I will be the moderator for today's session. Ladies and gentlemen, this webinar series was initiated with an aim in mind to share knowledge covering the Biomedical Museum of IMR. Today, we have with us the head of unit of Biomedical Museum and a very experienced assistant curator who both will share their experience and knowledge with us related to today's theme. Before we begin, please allow me to announce a few housekeeping messages from the Secretariat. We will not be taking any questions during the presentation, but participants may ask questions using the link provided in the email and YouTube channel. Q&A session will be conducted after both presentations. Attendance link will also be provided for those who would like to receive an e-certificate. Lastly, please subscribe to our National Institutes of Health YouTube channel to receive notifications. We will begin today's session by introducing our first speaker, Mr. Muhammad Hazilas. He was born in Perlis and started service with the government for over 22 years. He joined the Department of Museums and Antiquities in 2001 at the Maritime Archaeology Museum Unit, in addition to gaining experience with the terrestrial archaeology team by participating in several excavation projects in Sungai Mas, Kedah and Kota Tinggi, Johor. While working with the Maritime Archaeology Museum Unit, the Malaysian Museum Department received advice from Nanhai Marine Archaeology, which opened the opportunity for him to participate in the excavation of the Desaru Johor shipwreck, the Vanli Terengganu shipwreck, and the Tanjung Simpang Menggayau shipwreck Sabah. He also had the opportunity to gain experience with other divisions and units at the Malaysian Museum Department. For example, the Management Services Division from 2017 to 2014. Thereafter, from 2014 to 2017, he joined the National History Museum located in the Diplomatic Promenade of Precinct 15, Putrajaya. There, he gained new experience in the field of taxidermy of animals, birds, mold, casting, and bone articulation works. From 2017 to 2019, he returned to the Malaysian Museum Department as an assistant curator and was placed in the Documentation and Research Department. In July 2019, he joined the Biomedical Museum as an assistant curator. I would like to call upon Mr. Muhammad Hazilas to deliver his talk on the background and introduction of the Biomedical Museum.
Okay, Assalamualaikum and very good morning. Thank you, Miss Moderator, for a kind of introduction. Once again, thank you once again for the participants out there for joining us today in IMR-SEAMEO webinar. Okay, today I would like to present about background and introduction of Biomedical Museum. Okay, the Biomedical Museum, IMR, is located at Jalan Pahang, just nearby from the General Hospital, Kuala Lumpur. Okay, the Biomedical Museum was opened by YB Tan Sri Chong Hon Yan, which is Minister of Health Malaysia on 1st of June 1983. Okay, the aims to provide the information on IMR research activities and contribution to the scientific community. Okay, it's equipped with various collection of specimen and artifacts related to medical and health research collection since 1900. Okay, if you can see that picture, that buildings. Okay, there is the main building for the Biomedical Museum. Okay, IMR Biomedical Museum is located in the main block of heritage building, which was gazetted on 14th of April 1983. Um, it consists of two floors, uh, the ground floor and the first floor. Okay, for the first floor, we have a gallery, Malay traditional medicine and treatment. Okay, Malay traditional medicine and treatment gallery is situated at the ground floor. When you enter the buildings, okay, this is the first gallery you can see. This gallery collects and exhibits the equipment used in traditional medicine in Peninsular Malaysia. The exhibition has 5 sections, namely, first, herbal preparation. Second one is midwifery, and then followed by traditional Malay massage, shaman treatment, and cupping treatment or bekam. Okay, this is what we call as a mini segment, which located. We put this showcase at the stairs before you see the first floor. You're going to meet this showcase. The first one is flying squirrel and rat specimen, and then a bird specimen. Okay, when you reach at the first floor, you are going to see Dr. Wu Liante gallery. So in this exhibition, they have we exhibit the history, life experiences, photo album collection, achievement and contribution of Dr. Wu to the world. Okay, Dr. Wu was the first Malayan nominated for the Nobel Prize in psychology or medicine in 1935. Recently, we applied and are waiting for the approval of Dr. Wu's album collection on the plaque from the Department of the National Heritage as a heritage object. We hopefully can get as soon as possible. Okay, next gallery after Dr. Wu Liante gallery is a historical gallery. Okay, inside the historical gallery, we exhibit the establishment history of IMR and its achievement with commercialized products and innovation awards. This exhibition also exhibit various types of medical equipment and several types of human organs. Okay, this is the Snake Gallery. Snake Gallery is next to the Historical Gallery. The Snakes Gallery was opened in 2019 to the public. This gallery aims to display a collections of rare snakes, venomous snakes, and non-venomous snakes. uh, this gallery aims to increase general knowledge about snake species, their ecology, exposure on snake bites, and the production of antivenom. Also, we have a rare and endangered animals gallery. Um, the Rare and Endangered Animals Gallery showcases a wide variety of endangered animals in our country. Uh, the data obtained is from the IUCN Red List and our own specimens. The endangered animals on display are um, kokang, white-handed gibbon, western tarsier, a large flying fox, binturong, flat-headed cat, and many more. Okay, we also have a maggot debridement therapy gallery. Uh, the maggot debridement therapy gallery describes the historical process and success stories of a natural wound healing research, especially for diabetic patients. Also provides public with the insight and benefits of the sterilized Lucilla cuprina blowfly larva in wound healing. It also showcases the diabetic world treatment via educational and promotional videos. Okay, this is our latest gallery, but this is not ready yet. So the birds gallery, actually this gallery, we plan to move if you remember the bird from the stairs, from the mini gallery, we bring it here. Yeah, so it will comfort for the next visitors to visit this gallery soon. So the bird gallery exhibit various type of birds found in Malaysia. So among the birds on the display are dove, kingfishers, hornbill, watercock, sunbird, owl, eagle, and more. Okay, this is coming soon. Okay. Okay. We also have a repository for dry specimen. This, if you can see it in the slides, the dry specimen room for the Biomedical Museum. Uh, okay, in this picture is for dry specimen only. It's estimated specimen collection 5,000 plus of wet specimen and 10,000 plus of dry specimens. Biomedical Museum IMR has the largest rodent collection in Malaysia also. Okay, this is the wet repository for wet specimen. Okay, we go to an architecture and design. Uh, if you can see in the picture, if I think it's very clear, take from the air via drone. Um, this is main building for biomedical museums. Uh, the biomedical museum building is square shape. 1.5 floor is built from clay bricks. The roof part is made of clay known as marseilles, and there is also a flat roof part. Uh, for flat roof part, I think it's not very clear from here, but for the Limar roof, we can see very clear. So the architecture of this building is seen to be a combination of neoclassical and Art Deco. And Art Deco architecture also has characteristics that are influenced by mathematics and geometry, combined with nature forms such as curve from flora. Some classic features from the late Baroque architectural tradition. Neoclassical buildings can be divided into three main types. One type is the classical block buildings, and the biomedical museum buildings can be considered as buildings that resemble a classical block. Okay, for the interior, has repeating classical pattern of arches and columns. Overall pictures of the building is an classically decorated rectangular blocks and has a combined style of Art Deco architecture. Symmetrical and classic architecture design, carved with a combination of Art Deco and natural floral patterns. This is a picture for the interior design of the Biomedical Museum. Uh, Staircase, pillars. Okay, we'd like to thank you to Department of National Heritage of Malaysia for providing some information of the heritage building architecture. Okay, this is a few album for training and programs 2003. We did some conservation and preservation of natural history specimens. Uh, Basics introduction of zoological taxonomy, specimen digitization trainings, some album from the visitors who visit IMR Biomedical Museum. Okay, this is a Biomedical Museum visitor survey. Okay, how did you discover Biomedical Museum? Normally we will ask, and then we discover three top answers. The first one is social media, and then recommendation, and then the third one is website of IMR. Okay, this is the purpose of visit. The first one is for educational, second one for leisure, and the third one is for others. And then for the collaborations. Okay, if you can see here for visitors via from the country, we show the from Philippines, Indonesia, Singapore, show the top three of the visitors from overseas, followed by the other country. Okay, for visiting hours, uh, we open every weekdays from Monday till Thursday from 9 a.m. to 1 a.m. and then break for 1 hour, and then we open at 2 until 4. Just a bit uh, different for Friday, 9 till 12, and then we break for Friday pray, and then we open 3 to 4 p.m. We close for the Saturday and weekend, also public holiday. Okay, I think that's all. Thank you very much. Over to Miss Moderator.
Thank you, Mr. Muhammad Hazilas. Without further ado, we will continue our session by introducing our second speaker for today, Dr. Cheong Yun Ling. She was born in Negeri Sembilan and has served as a research officer in IMR for 20 years. Dr. Cheong first worked in IMR's Information Technology Unit for the initial 7 years and completed her master's in remote sensing and GIS in UPM. In 2010, she furthered her PhD studies in geography in the University of Humboldt in Berlin, Germany. She focused on the spatio-temporal study on dengue disease in Malaysia, vulnerability mapping, and environmental risk assessment. Her research interest is spatio-temporal epidemiology in infectious disease, including dengue, COVID, Zika, and TB. She served in the Epidemiology and Biostatistics Unit from 2014 to 2016, in the Medical Entomology Unit from 2017 to 2019, and Biomedical Epidemiology in 2020. She had the opportunity to gain research experience from various research collaborators from diverse backgrounds. Currently, she's working closely with the medical entomologists in the Wolbachia project, sterile insect technique, and insecticide resistance. She joined the Biomedical Museum unit in 2021 and is now the head of the unit. She extends her collaboration with other biodiversity groups. I would like to call upon Dr. Cheong Yun Ling to deliver her talk on biodiversity and geospatial research.
Thank you, Ms. Shamini. Hi, good morning. Today, I would like to bring you through the biodiversity and geospatial research in the Biomedical Museum, IMR. Well, I group it into two types, biodiversity and the geospatial. We conducted a lot of biodiversity research, and for this webinar, we present the selected one, MyBeast database projects, flying squirrels, studies on the bats, snakes, and studies on the publications of prominent zoologists. Let's start with the MyBeast. What is MyBeast? This is the Malaysia Biodiversity Information System database project that we collaborate with the Ministry of Energy and Natural Resources. Currently, up to date, we have already contributed more than 10,000 dry specimen records in the system. And you are welcome to search our specimens records online in this link, www.mybis.gov.my. And across the animal collections, we have the rodentia as the most highest specimens. And we also conducted studies on the large flying fox and the island flying fox. We compared these two species. We measured the head and body length, hind foot length, and etc. And we found that the males are larger than the females. And another morphological studies we conducted was on the Selangor pygmy flying squirrels. This is a special species. Why do I say it's special? Because it is only found in Selangor, and that's the reason why we name it as Selangor pygmy flying squirrel. And IMR do keeping 5 specimens on this species. And if you would like to know more about that, it is published in the IMR journal. And also we studies on the snakes. It's about the bibliometric analysis of the 69 years of publications on snakes. And the top author was the late Dr. Lim Buliet, followed by the Dr. Stephen Ambu. And we can see from the graph over here, the number of publications is keep on increasing, especially after the opening of the IMR snake farm and the launch of the anti-venin projects. And also, we conducted a biobibliometric analysis on the scientific papers of the late Dr. Lim Buliet, our prominent Malaysian zoologist. And this project is to profile the late Dr. Lim Buliet. He was a researcher in IMR and also a consultant of WHO. And we managed to extract 314 articles. We found that in this network graph, it shows that for most of the keywords, the publication that he published is about rats, mammals, small mammals, and rat lungworm. And the graph over here show that even after his retirement, he continuously published article and collaborated with other agencies. And this shows that our collaborations with the other ministry, like Ministry of Energy and Natural Resources, and also Ministry of National Unity. And we also exhibit our specimens in the Aquaria KLCC, DBKL, KL Dulu events. We participated in the event, and also we in the NIH Innovation Research Day. And the students are welcome to do the research on the specimens in IMR. Based on the specimens that we have, we already welcomed the students from IIUM, UITM, UMK. And we would like to take this opportunity also to welcome more students to come to study the biodiversities on the specimens. Well, let's talk about the geospatial research. These are some of the projects that we collaborate. The field release of Wolbachia, infected mosquitoes, insecticide resistance of dengue vectors project, COVID-19, dengue, Zika, nutrition, and TB projects. And there's our collaborators. We grouped it into a few techniques of the geospatial. And there's a list of the research questions or technical questions that we might ask when dealing with the geospatial research. Let's start with the mapping. Well, if we have the data, the research data, we might ask, how do I show my point in a map? We categorized Zika virus status by colored symbols, as you can see from this map. This is the base map is Google Map, and over this is OpenStreetMap. This is how we mapped the coordinates, and also we did the mapping for the categorization of the insecticide resistance level by colored points, different color of the red, yellow, and green represents different level of insecticide resistance. You can check our publications for further information. Well, the next question might be how to show the study sites in a map. We can even map our study sites based on the districts like this, or even without the Google Map, we can obtain the digital copy of the map from the Department of Surveying. And even so, we can also draw the map with the GIS software. One might be asking also how to show the rates by aggregation level in a map. Let's say for this study, we showed the categorization of the COVID-19 incidence rates by district level with the chloropleth map. Now, from the publication, we might move to the mobile apps. For the user convenience, we try to map for the location of the OV trap by using mobile apps. This is a collaboration project with the medical entomology unit and the research management unit. And also, we conducted this project by mapping the points of the study sites in the GIS system. From mapping a point, a district level, a polygon, we could also show a process in a map. Let's say for this study, we mapped the process of the spatial spread of the COVID-19 cases into Malaysia. So you can look at this, our Malaysia map, and China, Singapore, Cambodia, Thailand. It shows the initial process of the COVID-19 cases imported into the Malaysia. From mapping, we further discuss on the georeference. This is one of the technique of the GIS, the geospatial, how to digitize a paper building map. Let's say we have no information at all for the ground level. We could even obtain the technical drawing from the joint management body of a locality. Let's say for this map in Mentari Court, we actually did extract the technical drawing and then we map it with the GIS software and overlay with the Google Map. This is called georeference. What about spatial analysis? One might ask, how many outlets within buffer of 1 kilometer from house of the patient? We conducted a study to study the relationship between the availability of fast food outlets that were in close proximity to residential areas and overweight Malaysian children. Some of the techniques that we use includes web scraping coordinates of fast food outlets. We scrape it from the online, and we did the buffer analysis and points in the polygon. And the data we pump it into the multivariate logistic regression. And this is our findings. At least one fast food outlet within 1 kilometer from the reported residential address was significantly associated with overweight among children aged 5 to 18 years old. I'd like to show another project which uses the spatial analysis. What is the distance from the location A to location B? We applied it into the TB project to measure the distance of the house, the patient's house, to the nearest clinic. And the techniques over here, we geocode the patient's house and the hospitals and the clinics. We get the coordinates and then use the distance metric too, and also use the statistics to get the results. Spatial clustering. We might ask, where is the high-risk cluster? Often in a public health study, we like to know the spatial clustering. Let's say for this project, we would like to search for the spatial clustering of dengue cases. So we use the method of scan statistics to do that. And across the time, we can show the maps. And also use the spatial autocorrelation by district level using the local Moran's eye. The red color represents a higher clustering. The blue color is lower. What about the spatial interpolation? For this technique, we would like to know how to estimate the value for the no sampling location. As you can see from this is the spatial interpolation of the frequency of Wolbachia infected mosquitoes. We have the value for the black dots over here. This is our orbit trap location, but we do not know the rest. So we use the spatial interpolation to map it, to study. The technique that we use is the ordinary creaking and the spatial modeling and prediction. We can even predict dengue based on neighboring land use. We predict the occurrences of dengue cases using fishnet grid cells and boosted regression tree. So this is our final map of the prediction. We divide the grid by 200 meter times 200 meter spatial resolution. Our way forward. Research collaboration with more biodiversity experts on our specimens. And also, we would like to take one step further to digitize the specimen records and images. And also, bibliometric research on species, journals, subject areas, diseases, and prominent figures. Geospatial research for ecology and public health study. And we also welcome student attachment at museum. Lastly, we would like to expand our galleries. These are some of the selected publications in recent years, 2023, 2022, 2021 and 2020. And also selected presentations, consultation in recent years. With that, thank you. I would like to pass the time back to our moderator.
Thank you, Dr. Chong. We will now open the floor for a Q&A session. Okay, question to Dr. Chong. Do we have to pay to visit the museum? And what are the opening hours, please?
Thank you, Shamini. The museum is open for free for the moment. Welcome to visit museum. And the opening hours is already mentioned. You can get it from the website.
Thank you, Dr. Chong. Okay, a question to Encik Hazilas. What is the oldest item available in your museum? From Mr. Noah Ashraf.
He asked for the oldest item. Okay, actually I'm not really sure about the oldest item. If I'm not mistaken, I think the specimen of the birds.
Okay, thank you, Encik Muhammad Hazilas. Question to Encik Muhammad Hazilas. Are there any special activities organized from time to time?
Okay, actually, there is no activities, but it's upon a request, and it depends on how many. For example, if you bring the kindergarten kids, maybe we can create some activities, the suitable activities. But you have to be request maybe an email. The state mentioned that you want to do some activities, so maybe we can try to create some activities for the visitors.
Thank you, Encik Muhammad Hazilas. Okay,
Wow.
question to Dr. Chong. Interesting museum. Thank you. Do you plan to have special video, nice photos, comprehensive website on your building and collection? This question is from Puan Hana Nurin.
Thank you. Oh, it's a very good suggestion. We will think about that in the future. Thank you.
Next question to Encik Muhammad Hazilas. If there's a big group visit, how would someone conduct it?
Okay, normally we are going to split by 3, 5, 3 to 4, 5 group. It depends on the total.
Thank you. Okay, question to Dr. Chong. Okay, it's from Puan Ashura. I'm a school student. Can you please explain why we need bibliometric analysis?
Thank you for the question. The bibliometric analysis is actually a text analysis, and it's looking at the publication over the years, because let's say for a certain topics, let's say for COVID-19, we can do the bibliometry on COVID-19 over the 2 years, and if we would like to know who is the most leading author, what is the main subject study, and also which institutes published the most about COVID-19.
Okay, thank you, Dr. Chong. Okay, last question for Cik Muhammad Hazilas. What about the parking in the Biomedical Museum? Do you have ample parking? Do we have to pay for parking?
Okay, nowadays there's a lot of parking lot, but for the bus, I think it's a bit limited. But no need to the it's a free parking.
Okay. Okay, thank you.
Okay.
Okay, I'm afraid that's all the time we have for Q&A session. Any additional questions will be answered by the speakers through email. We hope that this webinar has shed light on the Biomedical Museum of IMR and would spark interest and could initiate research collaborations with others. Ladies and gentlemen, now we have come to the end of this webinar session. I would like to thank all participants for their support and for making the session successful. Don't forget to subscribe to our National Institute of Health Malaysia YouTube channel. Before I conclude, I would like to announce that we will have the next session of IMR-SEAMEO webinar series on the 22nd of September 2023. So mark your calendars, please. The theme for the next webinar is mesenchymal stem cells and other preclinical models for cancer therapy. We hope you can join us at the next session. Until then, many thanks from all of us at IMR. Take care and stay safe.